What I wanted to do is to review the in-class notes that we did, so I'm giving an abbreviated version of what we did in class because I didn't record the MP3s for the entire class. So on your in-class notes, the PDF that I have of that, we went through and worked through slide 14, the renin-angiotensin system, aldosterone system, so that's RAAS, it's listed here, and it corresponds to this slide that we have here for the renin-angiotensin-aldosterone system. And there's a couple of things that we're pointing out here. The major feature that we're looking at is how a drop in blood pressure can cause a whole series of changes to help and work to increase blood pressure and blood volume. And we worked through a lot of these different things in the MP3, but what I wanted to do was to be able to help and reinforce some of these different principles. And in particular, going back to our PDF that we have, what I asked people to do is to identify mean arterial pressure and the three variables that are important for it. And so these parts here, stroke volume, heart rate, and total peripheral resistance were blank. And I asked people to identify what's a major equation for the mean arterial pressure that involves these three factors. And then I asked them to identify where in that diagram on slide 14 each of these things is corresponded to roughly. And so for what this is trying to emphasize is that our arterial constrictions that we have in the diagram relate to total peripheral resistance. And so going back to our diagram, here it is. Here's our arterioles that are constricting in the presence of activation of the renin-angiotensin system. So renin's released here, and then converts angiotensin into angiotensin 1, and that in turn, angiotensin 1 turns into angiotensin 2 when it's presented with ACE, this angiotensin converting enzyme. That in turn then causes vasoconstriction of the arterioles, and that elevates our blood pressure. Great. So that's our TPR. And then as far as stroke volume and heart rate, those two things are regulated by our cardiovascular control center. And so then we went into and talked a little bit about the way that that's modulated. And I'm going to scoot down here and show you this region here, which emphasizes how heart rate, stroke volume, and TPR are regulated by our autonomic nervous system. Norepinephrine from our sympathetic nervous system can act on beta-1 receptors in the autorhythmic cells to control heart rate, beta-1 receptors to activate our stroke volume, and also act on alpha-1 receptors in the arterioles to increase our total peripheral resistance by vasoconstriction. And so I emphasize that these were things that could come up on the final exam. It's kind of an integrative type of problem. And so they would need to be able to, you need to be able to be familiar with these different components. The receptor subtypes, norepinephrine, and how these different branches of the autonomic nervous system regulate these features of mean arterial blood pressure. And the parasympathetic system is acting through acetylcholine on the muscarinic acetylcholine receptor on the autorhythmic cells, but it doesn't have a direct effect on stroke volume or total peripheral resistance. So the other thing I said is I had a little bullet point list here of some other things that are potential target points for the final, including the type of action potential that we get out of the autorhythmic cells versus the contractile cells, whether they contract or they don't contract, thinking of autorhythmic versus the contractile cells, the equation of end diastolic minus end systolic volume equaling to our stroke volume here, and then finally our mean arterial blood pressure, an alternative way of calculating it. And so those are things that are fair game for the final, and so I emphasize them here. So going back into this then, we have a second question that comes up is what if you block the ACE enzyme with a drug, what would happen to mean arterial blood pressure, what would happen to sodium retention, and what would happen to water retention? And the answers are posted here that we came up with, but the slide that we have that we're going to refer back to is here. So the thing that we're blocking is that we're blocking the ACE enzyme that's responsible for converting angiotensin 1 into angiotensin 2. With blockage here, what's occurring is that we've made it so that this pathway that's all about trying to bring our blood pressure back up is now being blocked. And so we're going to keep our blood pressure low because we're not going to be able to cause vasoconstriction to happen as much. We're not going to be able to induce as strong of a cardiovascular response. We're not going to be acting on vasopressin or antidiuretic hormone to help retain water. And so vasopressin is all about water retention. And we also aren't commanding the release of aldosterone to help promote sodium retention. And so all of these different things, vasoconstriction, cardiovascular response, vasopressin, and aldosterone, are all regulated by lots of different things outside of renin. But renin's role on this and effect on the tone of these different factors is affected, so that you end up having less vasoconstriction, less of a cardiovascular response, so drop in heart rate, drop in stroke volume, less vasopressin released, and less aldosterone released. So you lose more of your of water, and you have less water retention, and you also have less sodium retention if you block this ACE enzyme here. And so that's a therapeutic agent that's used to help regulate blood pressure. So that talks about some of these major features here. The other thing that we did is we went back and brought it back to slide number 15 that talked about aldosterone and went through all of these different steps here. These are outlined well in the slide and also in the MP3. The major thing that we focused on and helped reinforce was not only the sequence of steps here, but to approach it from thinking about, if you want sodium retention, you need a sodium-potassium pump. And you want to position it here on what's known as the basolateral side. So if you think of basolateral, basement membrane, where your feet are closest to, the feet of the cell are sitting on top of the basolateral membrane, and the apical side of the cell is reaching into this lumen of the nephron. And so apical is on this side, and basolateral is on this side. And so those are two features that we want to try to illustrate here. The sodium-potassium pump is responsible for this main action of aldosterone, and the way that it works is aldosterone promotes the transcription of more sodium-potassium pump, and also inserts channels to permit the influx of sodium here, and also the efflux of potassium out as a consequence of the action of the sodium-potassium pump. So in these cases, what you have is more sodium being taken out and more potassium being excreted. We also went and visited the action of vasopressin here, and just made sure and reinforced this idea of where vasopressin is acting, and that it's acting through this mechanism, through a second messenger system, and also through aquaporins. So that concludes part one of our descriptions, and I'll do on a separate movie the part on the next PDF slide.